So we're going to talk about blood. It's a transport vehicle in the cardiovascular system. Carries oxygen and nutrients to the tissues from the heart. It takes the oxygenated blood and collects wastes from the tissues and distributes blood in a way in a pulmonary circuit so the blood can get oxygenated. Also prevents the leakage. It contains coagulation system that prevents the blood loss. It plays an important role in the heat exchange in the skin. Blood consists of plasma, and so this is plasma. This is formed elements. Plasma essentially is the fluid, mostly water, proteins, electrolytes, nutrients, and wastes. It helps to maintain so-called osmotic pressure of the blood. Formed elements of the blood are cells. They all originate in the bone marrow, in the red bone marrow. There are three main types. There are red blood cells that deliver oxygen. There are white blood cells. It's the huge group here. They protect the body, provide immunity. And platelets, which are responsible for clotting coagulation. So there are 5 types of white blood cells that you need to know. 3 types belong to the group called granulocytes. It's eosinophil, basophil, and neutrophil cells. And monocytes and lymphocytes are agranulocytes. So you have granulocytes here, agranulocytes here, and each type is divided further. So let's talk about white blood cells a little bit more. So neutrophils, basophils, and eosinophils. Eosinophils protect you from infections with various eukaryotic pathogens, protozoa, helminths, so on and so forth. Basophils are responsible for inflammatory responses. The neutrophils here protect you against bacteria. Lymphocytes, and these 2 cells here are agranulocytes. So lymphocytes protect you against viruses and cancer cells, while monocytes are the main phagocytic cells in the blood. They eaters, big eaters. So red blood cells transport oxygen to the tissues. They are the most numerous blood cells. In the blood cell, a red blood cell, there are many molecules of hemoglobin, and each molecule of hemoglobin contains 4 molecules of organic compound called heme. Heme, in turn, in the center has an atom of iron. Oxygen can bind to iron and can be delivered to the tissues. Essentially, 1 molecule of hemoglobin can carry 4 atoms of oxygen. If 1 red blood cell has 200 million hemoglobin molecules, 1 cell can carry about 800 million atoms of oxygen. They live for about 4 months, and then they are removed. The red blood cells are removed from the circulation by spleen and liver, and we produce them, and we destroy them, and produce and destroy. And the process of generating red blood cells is called erythropoiesis. Here you can see the scheme of erythropoiesis. Basically, when you are in need of more red blood cells, let's say because of the oxygen shortage, your bone marrow will receive the signal from the kidneys to start producing more red blood cells. You're well aware of so-called blood types. So blood types, there are many blood types, first of all, but the blood group that we're going to be talking about is so-called ABO blood group. And it is described by the presence or absence of certain markers on the surface of the red blood cells. So for instance, blood type A contains A antigens or agglutinogens, okay? Those are markers. These markers are agglutinogens on the surface of the red blood cell. Type B contains B agglutinogen. Type AB contains both A and B agglutinogen, and type O contains nothing. No agglutinogens at all, no markers. And in the plasma of people with each blood group, there are antibodies that can bind to these agglutinogens. Antibodies that are called agglutinins. When antibodies bind to the agglutinogens, they lead to clumping, like here or here or here, clumping of these red blood cells. So the blood of person with type A contains antibodies against B. Blood type B contains antibodies against A. Blood type AB contains no antibodies at all. And type O contains both anti-A and anti-B agglutinins. Which essentially, if you think about it, when we transfuse blood, we mostly focused on transfusion of red blood cells. So if you want to transfuse, the best option is to have either matching blood types or have a blood that has no markers at all, because in type O there are no markers. You can put this blood type into any of these groups, and they agglutinins have nothing to clump. So that makes a person with type O a universal donor. On the other hand, if you look at the person who can receive any type of blood, that's type AB. Why? Because they have no antibodies. Their plasma cannot agglutinate anything. So type AB is the universal recipient. Now, lastly, we're going to talk about the platelets. So platelets are very small cellular fragments, sort of, big cells called megakaryocytes produce platelets. They do contain elements of the cell like mRNA. They don't have DNA, they don't have nucleus. They do have mRNA, proteins, and enzymes. When platelets are activated, they form a clot. Not only this, damage to the blood vessel leads to the platelet activation. You can see red blood cells trapped here. Platelets are too small to be seen. Platelet activation and initiation of coagulation pathways. Coagulation pathways, there is an intrinsic pathway, extrinsic pathway, and intrinsic pathway. Both of these pathways lead to the formation of a fibrins. You probably can see here at the bottom the green threads shown here. So these threads are fibrin threads. They essentially enforce a thrombus, make it harder to destroy. The difference between extrinsic and intrinsic pathway is what activates it. Intrinsic pathway, which takes more time, can be activated by the trauma that is contained inside of the vessel. You see, vessel itself isn't damaged. It can be damaged to endothelial cells or some other kind of activation inside the vessel. Extrinsic pathway it's much faster, and it's initiated by some kind of a damage to the tissue rather than endothelium or blood vessels, like in case of blood cells, like in case of intrinsic pathway. Lastly, we're going to say a few words about blood disorders. So the most common, of course, is leukemia. It's cancer of bone marrow. You can see here abnormally shaped white blood cells. There are different types of leukemia. There's acute leukemia, chronic leukemia. In acute leukemia, you have many absolutely immature white blood cells, and the chronic, which runs longer. Acute is can end the life of a patient much faster. So chronic leukemia cells are more developed, but they're still not fully mature. Another condition is anemia, when you don't have enough red blood cells. So patients with anemia will experience things like weakness, shortness of breath, and fatigue. Anemia can stem from iron deficiency, okay, when there's not enough dietary iron. Anemia can stem from the shortage of vitamin B12, which is important for fast dividing cells. This is called pernicious anemia. And anemia can stem from the problems with the bone marrow. When bone marrow cannot produce enough red blood cells, it's called aplastic anemia. And of course, hemorrhagic anemia. Somebody bleeds, but that's restorable, basically. Okay, hemorrhagic anemia is restorable. Now, lastly, I want to say a few words about sickle cell anemia. It's an interesting condition. In sickle cell anemia, patients have a mutation in one gene for hemoglobin, and they have one amino acid change from glutamine to valine. And that little change leads to the deformity of the red blood cells. They become shaped like a sickle here, you can see on this bottom image. These sickle-shaped cells cannot pass through the vessels, which damages the endothelium and increases the hemolysis. Low number of functional red blood cells leads to hypoxia in tissues. And the similar condition as thalassemia. In thalassemia, patients usually have lower content of hemoglobin and fewer red blood cells. And here you can see a typical patient with thalassemia because it's associated with multiple developmental abnormalities like deformities of the bone or heart problems. What is very interesting about anemia and thalassemia is that patients with sickle cell anemia or patients with thalassemia cannot be infected with malaria. It's actually a very interesting map here. So on the left, you can see the distribution of sickle cell gene in Africa, and on the right, you can see the distribution of malaria. Essentially, sorry about that, essentially, that's the evolutionary advantage, paradoxically, of having sickle cell anemia gene, not necessarily disease. You can have one copy of the gene. You're going to get sick if you have two copies. If you have one, you're not going to get sick with sickle cell anemia, and you're also not going to acquire malaria.